plan for today is to go through a bit of theory, given the interaction, and then expose you to a lot of terminologies that's actually specific to RNA-seq analysis, and then give a bit of a tutorial for today, and then probably by the next module we'll have a more in-depth tutorial and everything. So so the thing is, initially there are two different kinds of RNA-seq analysis. So one is what we call as a bulk RNA-seq analysis, and other is what we call a single cell RNA-seq analysis. So the difference between two is that here in bulk you have several combinations of cell. For example, if you want to characterize a tumor that is present in your body, and you want to take the cells. So in case of bulk analysis, there is this kind of analysis has got several cells from the same tissue. So you can very well capture the heterogeneity of the tumor and everything. Whereas in case of single cell RNA-seq, a single cell is isolated, like it is isolated, and then only that is PCR amplified, and then sequenced, and a bunch of analysis are done on those. So today what we're going to discuss is is a bulk RNA-seq from the viewpoint of a bulk RNA-seq analysis, because this is what I've been dealing with, and then probably in the next class I can give you like a very brief introduction to RNA-seq, single cell RNA-seq, because there are very few there are differences in the statistical viewpoint. Obviously there will be differences, so I could cover that in the next workshop. So yeah, so what is RNA-seq? So this is a technique or a method in which you can quantify the gene's expression at a particular state of at this particular state of the cell. Like you can quantify the gene expression, for example, with respect to expression of a tumor cell with respect to a normal cell. Hello, so Mahesh, so I think you could mute your mic because there is some background noise.
It's not mine, it's some of the participants.
Okay. So yeah, so this this is actually a way to quantify the gene expression. So for example, if you want to look at the presence of a few genes that is actually being upregulated at a specific time point, or downregulated with respect to another cell, RNA-seq is the way to go. So it is like trying to analyze the gene expression at different states, like different states of the particular cell. And then actually before RNA-seq, this was actually done through a lot of methods. For example, if you want to quantify a gene expression, what the general way would be to tag it with a fluorescent tag, and then look at it, look at the strength of the fluorescent signal, and then you kind of have an idea about what would be the expression of this particular gene. And there have been several other analyses, such as SAGE analysis, and then a lot of other analysis for you to quantify gene expression. But this all led to the microarrays, which were actually used for a very long time. So when you want to look at this kind of differential expression analysis, like when I talked about, you want to see which what are the genes that is being upregulated or downregulated in a tumor cell with respect to any normal cell that is in a normal state of cell. So all this analysis was actually done using microarrays. So the problem about microarrays was that you should really have a pre-information regarding the gene structure and the gene, the information about the gene. Only then you will be able to check out, because it's more like hybridization. You would want the gene sequence, everything, and then only then you could actually look at the expression level and everything. So, but then when NGS was happened and everything, so this led us to look at the transcriptome of a cell without having any prior gene information. So the idea is that you take all the contents of a cell, you remove the DNA, you remove every other that you deem is not actually suitable for your transcriptome, and then you and then you isolate only the transcriptome, and then you try to sequence it using. And so after isolating the transcriptome, you actually create a cDNA library, you convert into DNA, and then you try to sequence that. So this would give us an idea about the transcriptome of that could be captured from a cell. So this could actually, the best part is that even if you're using any other type of analysis, like for example a microarray, again there is this problem of transcript. For example, you don't know which particular transcript is actually being expressed in a particular condition, or which particular transcript is favored by the cell and everything. So these transcripts can also be captured by using an RNA-seq. And RNA-seq, there are few other very main this could also be very main sources of information, such as fusion genes, for example. So I mainly learn everything regarding to tumor in my work. So when you look at fusion genes, they are like a very main source of drivers of tumors. For example, what happens in fusion genes is that a part of a gene from one like few parts of a gene from one gene actually fuses with another part of another gene. So this the underlying event is actually a structural event. For example, it could be a deletion, it could be replication, translocation, or an inversion. But the best part is that these RNA-seq, since it is a transcribed version of the structural event, could give us a really good idea about these particular fuse genes. And there have been many fuse genes that are actually very known, like they're known drivers. For example, in case of the ALL cancer, that is the ALL cancer, that it's a type of leukemia actually. These leukemia, in case of those kinds of cancers, you have several the cancer itself could be subclassified based upon the fusion genes that has occurred in them. So this is RNA-seq is actually a very good source to learn about that. But obviously the validation has to be done by using any other, for example, you want to go even more downstream and you want to kind of validate with the proteomics analysis and everything. So, and in case if you don't have a DNA material available or anything, for example, you lost the sample and you want to do a variant analysis and everything. So this could actually be a very good source for variant analysis. There's also this, given the fact that there are many variants, there are a lot of modifications that occurs after transcription. Given that, even given all those information, we can still, with a good likelihood and with good probability, we could predict variants using RNA-seq. So that is also another use of RNA-seq. So what I put here in this slide is actually the general workflow that everybody mostly it's done in RNA-seq actually. So the idea is that we get RNA-seq FASTQ files after sequencing. So this sequence FASTQ files are either aligned to a reference genome if the reference genome is available, or we could even align these reference genomes to transcriptomes. So we have several transcripts that is available, and then you can even align it to specific transcriptomes. Or in case of two conditions. For example, if you don't have any reference information for this particular organism, not readily available, or their annotation is not readily available, so you could actually go to a de novo assembly, and then you can quantify. Or if, for example, you want to discover new transcripts that's already not present in the in the annotations. So in such a case, you would again want to do a de novo assembly. So as you proceed down from that particular level, so we'd have to do a bunch of quality control. So these quality control occurs at different levels. For example, at the FASTQ level, and then you look at the alignment file, and then you do a set of quality controls. And then there's this other step called as the exploratory data analysis. So here again, this is also like a quality control per se. Because for example, you're looking at looking at two different cells. One cell is being controlled, like source of control, and the other cell you have grown in a particular condition. I'm just giving examples here. And you do a bunch of exploratory data analysis, and you look at a PCA, and you look, and so for example, there might be few cells that have been cultured under a given condition that might group with the control samples. So in such cases, we would want to remove them. So exploratory data analysis is something that validates your hypothesis that okay, this particular condition, or this particular perturbation has actually changed the cells, or you could actually visualize it, and then again that can act as a quality control. So, and the main use of RNA-seq is mainly to look at differential expression analysis. So this differential expression is nothing but you want to call a bunch of genes that are either very highly expressed than the normal, or you want to either they're very highly expressed or very low expressed. But the idea is you want to call them with a very good probability that they could be a true positive. So this differential expression analysis, there are several tools which actually use different kind of modeling and a lot of stuff. So today we'll be covering only into the exploratory data analysis, and then a bit of differential analysis, because I'll be using the tool that I generally use for differential expression analysis for the exploratory data analysis. And then the idea is that once you get a bunch of genes, for example, you get a bunch of genes that are upregulated and you get a bunch of genes that are downregulated, and then you would want to see what pathways are actually present in these genes. So then what we actually perform something called as a pathway enrichment analysis. So these pathway enrichment analysis, again, you have several resources to do it from and several tools, and then there's several ways to look at pathway enrichment analysis. There are also networks. There are network topological analysis also, that there are few of them that could give you a very good idea of hub genes and all that. So I'll be covering it in the next class. So yeah, so I'll start with how these RNA-seq libraries are actually obtained and then used. Okay, so the first step would be to isolate all the RNAs. So there are two kinds when you create an RNA-seq library. One is a total RNA-seq library, where you have ribosomal RNA, mRNA, tRNAs, and several other small RNAs that are generally present. So and you also look at mRNA. So mRNA is something that gives you the expression data. So this is mRNA is the only one that is actually upstream to the proteins, right? So if you want to typically look at expression data, such as you want to look at the genes that are involved in or the pathways that are involved in, that is responsible to cause this particular condition. So then we do a mRNA analysis. So, but there are analysis where you look at very small RNAs like we interacting elements and then microRNAs. So those are very highly specific, and those where rest of the other RNAs have to be depleted and they have to be for the use. So I'm going to stick with the widely used mRNA-seq analysis, because that's the one that's widely used. So like I said, your first step would be to remove all the other contents that is in the cell. We would only want to isolate RNA. So within RNA, about 90% of the RNA is composed of ribosomal RNAs. So if you want to look at only the mRNA, so there are two ways in which you could actually obtain mRNA that is being used very widely. The main way is to have a poly A tail selection. So we select using a poly A tail, and only those fragments are, they are amplified and they are further sequenced. Otherwise, there's another way to do this is to deplete all the rRNAs. That is generally called as rRNA depletion method. So these two methods are mainly used, and libraries, sequencing and then alignment, quality, everything is actually better when we use a poly A selection than a rRNA depletion method. So like I said, I've given a brief summary of that over here. So so I'll be going with mRNA-seq and then for the rest of the examples. So yeah, so the next step, what we would want to do is, after you have the FASTQ files and everything, you would want to after you have the FASTQ files, you want to check out if the particular organism has got a good reference. So our main idea the next step would be to have a good alignment and quantification. So the quantification here is nothing but I'll be showing you an example of the read counts. So here quantification literally means read counts. So for every single sample that you're going to have, for every single gene, you want the number of reads that are actually mapped to that. So what these alignment tools over here do is that they give you these counts specifically. So these counts are actually only used for differential analysis, other pathway analysis, and several other analysis. So so there are many alignment tools that are specific for RNA-seq. So why do we want new alignment tools than what we use for DNA-seq is that, so for example, you have a reference. The reference has got all the, all the different structures, right? It would have introns, it would have exons, and then it would have 5-UTR, 3-UTR, and mainly, but in case of mRNA-seq and mRNA-seq, when you look at it, it would not have the introns per se. So they're all spliced and only the exons are present. So we want splice-aware tools. So you want these tools to be aware that there are few portions in the reference genome where these reads won't map to. So these kinds of tools are called as splice-aware tools. And the most used one right now is the STAR tool. So there's STAR and there is Top Hat. And but the main thing is that these tools have very, very large footprints. Excuse me. Yeah. So these STAR and Top Hat, they have very large footprints. For example, they create an index of your entire genome, and then it requires a lot of memory. For example, it would at least require 90 GB of memory, not memory, it would require 30 GB of RAM and everything, but it would actually require like around 90 GB of space where it can store a lot of intermediate files and everything. So STAR is a very computationally expensive and very resourceful tool. But the best part is that STAR can also handle non-trimmed FASTQ files, because all these intron regions that you think are introns are actually soft clipped by STAR. And the same is done by Top Hat. And HiSat is actually one of the pseudo aligners where you can actually use, it can actually, there are pseudo aligners where you can actually use a very small RAM, for example, 4 GB. So they use a BWT algorithm, like Burrows-Wheeler transform, to actually create a complete index of the entire genome, and as well as create small indexes. So the first pass is done using the entire index, and then then the mapping is done through small indices. So it only takes around 4 GB of memory, and then you'll still be able to accomplish quantification and alignment in your laptop, whereas STAR and Top Hat you cannot do that. So like I said, all these Bouty and other DNA-seq aligners do not work here. Bouty, Bouty2, BWA. So these are not what you would want to align with when you're using a RNA-seq. So so this alignment can actually be done to a reference genome where you have, like, for example, it could be a complete reference genome, as well as it could also be a transcriptome, depending on what you want to look at. For example, you want to so for example, you want to have only gene counts, or you want to have counts that are specific to specific transcripts that you want to look at. And in such cases, for example, you want to compare which transcript of a particular gene is actually being differentially expressed and all that. When you have a very specific case, you can align it to a transcriptome. So star, top hat, I said all of them also allow alignment to a transcriptome. And there are pseudo aligners that are specific for transcripts. For example, Callisto, Salmon, and Sailfish. So these pseudo aligners, again, like I said, they do not completely align. It does not create a BAM file. What they do is they only give you these read counts that you want to use for downstream processes. So all of these are the choices that you would want to look at when you have a reference genome, as well as a very good annotation GTF. When the organism that you're working with does not have any of this, the best way would be to use a de novo aligner. So you can use a de novo aligner such as Trinity or Trans-Abyss. So Trinity is actually from the same group as Star, and they're a very good group from Canada. Like so it's they have the it's a very good group, and then most of the papers use Star or Trinity and everything. So that is my opinion over here. So the idea is that this quantification, the read count and everything would give you an idea about the kind of library that's been prepped. Like, for example, in case of Star, there is also this main, in case of Rna-Seq, there is this main idea called, while creating these, when you're, sequencing, so they can be unstranded, where you have a single strand analysis, and they could also be paired end sequences, where you actually, you sequence both the forward strand and the reverse strand. So, for example, when you're actually selecting by poly A tail, you are actually selecting, the mRNA, and then it is actually, it is converted into a cDNA, and then it is, the complementary strand is created and everything. So when you use in such a case, it is actually reverse transcribed. We call it as reverse transcribed because since it's a cDNA, you have it actually goes in the opposite direction. So the reverse, so Star actually gives you the counts based for all the three conditions. For example, if the particular, the kind of sequencing that you're done, the kind of library you prepare to sequence, all that, if it is actually an unstranded library, or a forward stranded library, or a reverse stranded library. So you have to actually correctly pertain, and then use the correct count. And you also have several statistical tools that actually looks at these counts and predicts which kind of library that was prepped. So that could also be a quality check. So these, and I also want to tell you that these read counts from the Rna-Seq analysis are actually, there's a repository for it in NCPA, and it's under the gene expression omnibus. So yeah, so let's come to quality control. So when you're looking at quality control, you have to start from the FastQC. So here there are few criteria that you really need to look into. For example, per base sequencing quality has to be good across all the bases. And if you see very bad quality in case of, like above, like if you're using a 150 base pair library and everything. So if you see bad quality control at 3' please trim it. Or like if, for example, if you're using an aligner such as Star, it would not matter because it will do a lot of soft clipping by itself. But if you really want to have a very good alignment, statistics, and everything, I would recommend you to trim those, like do a filtering based on the quality or something like that. So there are several tools to do these trimming for RNA-seq. Like we can also use cut adapt. That is generally used for DNA. And then there is skewer. There are a lot of tools that looks at adapter contamination, such as minions. So there are several tools for RNA-seq specifically. And then one of the main things that is very characteristic for RNA-seq is that it's got very high number of duplications and a lot of overrepresented sequences. That's something that we can actually expect, because given that we are not exactly looking at individual sequences that when we look at our DNA-seq. So we are actually looking at genes, the expression of genes. So already there must be several copies, and then there is, if for example, if one of the gene is overrepresented or it is highly upregulated, so we would want to see a lot of these gene, a lot of these reads in the FAST-QC. So so that is like kind of intuitive to not to look at high the duplication reads and the amount of duplication over represented sequences. So you would also want to look at GC content, because GC content gives you a lot of insight into contamination. For example, if it's a contamination of adapters, or there's some adapter ligation, and then there's a lot of adapters in the for example, in like when you sequence, it's there's a lot of adapter contamination. So the idea is that when you look at the graph of GC content and you look at the percentage of reads that's got this percentage of GC content. For example, if there are if it is a bimodal curve, so you have a peak at 10% and you have a peak at 90%. So that would clearly say that there is adapter contamination. It's like a lot of reads that's got 10% of GC, and then a lot of reads that's got 90% of GC content in their sequences. So like when there is a very high GC content, so it could also be, one thing is that it could also be the organism specific. So, but then when you're looking at human, for example, you would not want to see a bimodal curve. You would want to see a normalized curve. And you should actually do a bit of research to look at what kind of graph that you get for every, like if you're working with different organisms, you would want to have a basic research done. So because we cannot blindly use the same cutoff for different organisms. So, but then encode, and main thing is that, so in DNA-seq, we have depth, right? Like for example, when you, so depth would be called as the amount of times a particular base is being sequenced by the sequencing machine. So, but here in RNA-seq, the depth per se is not a thing. So because depth per se is not a thing is because when you look at coverage, you have a lot of coverage where the genes are highly expressed, and we would not have a lot of coverage where genes are not really expressed. So what the analogous to depth in case of RNA-seq is the total number of reads actually. So and there are cutoffs where you have to have at least these number of reads to do a proper analysis. But people have done a lot of, like people do with very, very less number of reads as well. But, to have a very good statistical power and everything, you would like ENCODE recommends, for example, around 30 million reads for mRNA-seq of human samples. But 30 million reads is a huge number of reads. So again, it also depends upon what kind of sequencing you're using and etc. And other thing to look at quality control is to look at the BAM file. So there are several tools such as QualiMap, RNA-seq, all of which are exclusively developed for RNA-seq. So these tools, what they do is they look at the alignment statistics. For example, they look at percentage of reads that are uniquely mapped, and then percentage of reads that are actually mapped to so since we are using an RNA-seq, you would want a lot of reads to map to the exonic region. You would want a lot of reads mapped to UTR, and because 3-UTR and 5-UTR still would not be spliced in case of mRNA. So So a lot of reads actually map to 3' and 5' UTR. And there might be some amount of reads that is mapping to intergenic regions and then other intronic regions as well. So the idea is that this percentage of reads mapping to intronic and intergenic regions, as well as if it is, for example, an mRNA-seq, where you have depleted the rRNA or you have done a poly selection, you would want very, very less number of rRNA, because RNA contamination is it's like very very recurring one in case of RNA-seq. Like if you want to look at mRNA-seq, you should have very, very like less than 5% of rRNA. So yeah, so all these alignment statistics give you a lot of information regarding the library preparation. And like I said, if there is a lot of contamination, you will have you can actually say that there is very so for example, again, if you have a lot of reads that are not mapped, so in star it gives you a percentage of uniquely mapped. So at least you would want to have more than 90%. If you've done a very, very good library, you have more than 90% of the reads mapping to the reference genome in uniquely mapped. They'll be uniquely mapped. Otherwise, there might be a lot of there might be some percentage of reads that are actually multi-map, because there are so many pseudogenes and repetitive sequences in our genome. So those could be the reason. And but if there are a lot of reads that are actually not mapped, that could mean that there is a contamination, because these reads are actually might be from another organism and it's not mapping to the reference genome that you've got. So yeah, so all of these information give you a good insight into quality control and where these problems have arisen from. And also you have several statistics to look at, for example, in star, mapped mismatch rate and then deletion rate and all that. So those could give you an insight into the sequencer quality, like for example, the sequencer itself is making an error or not. So these are the few criteria to for quality control. So yeah. So what we have to do is, like generally in RNA-seq, what we get a bunch of, like I said, there are different genes. For example, we have gene X, gene Y, and gene Z, and there are several genes that are mapping to it. So using this RNA-seq, what we would want to do is to compare. Either for one thing, we would want to compare it within the same sample. For example, this sample A, you would want to compare it within sample A, or you would want to compare between samples. For example, you want to compare gene X with respect to gene X in sample A and sample B. The problem is that we cannot directly compare. For example, when you look at sample A, you can see, and for example, for right now, do not consider gene DE. So if you don't consider this particular gene DE, so when you compare gene X across sample A and sample B, we can see a lot of reads. Like, sample A generally having a lot more reads than sample B. So we cannot, when you do a rough comparison like this, gene X from sample A and sample B, we can say that gene A is a lot more reads than sample B. It could be upregulated in sample A, not sample B. But the overall the overall coverage or the depth, as I see it, of sample A is greater because it's got very high number of reads compared to gene B. So what we want to do at this point is to normalize these counts. So once we normalize, we could actually compare within sample, between samples, or you can actually see if it is actually a differently expressed gene, or it is actually an anomaly that is being caused because of difference in depth in different samples. For example, I'm saying, so when you want to compare it between two samples in the same gene, it would make sense to divide it by the total number of genes total number of reads that are present in everything. So we cannot have that, because if we only divide by the total number of genes, And if you want to do comparison between gene X and gene Y of the same within the same sample A, then again there is a the length of the gene also matters. This also should be taken into account. So this particular length, this gene X seems to be larger than, like, significantly larger than gene gene Y. So this would give a lot more counts than gene Y. So normalization also has to be in account of the gene length. And also, when you look at these differentially expressed genes in the general genes, right? So once we account for the, so when you, if you, like, for example, if you consider this gene D right now, if you want to normalize for the entire sample A and entire sample B, this gene D, the differentially expressed gene has a very high upregulation. It's a very, very high regulation. So when you divide it by the entire number, like, it would give significantly high number if you add up all these reads. So this very, very high number would actually, when you normalize it, it would lead to a very small number of read counts in other genes. So we should also take this into account. So this is called as RNA composition. So where we also want to take into account where we have to discard these, these information of the gene DE when you're looking at, when you want to normalize it. So there are several different ways normalization has been done. It is started with the, like, very simple way where you simply scale it by the total number of reads. And then, but then the problem is that you cannot, like I said before, you cannot use it for a differential expression analysis because you do not account for, very high reads that are actually a result of the differential expressed upregulated or gene or a downregulated gene. So that is something you cannot do. And then you can also, the only thing you can do is to compare between two samples, and you cannot compare within the samples because gene length is not accounted for over here. Then came actually after this counts per million came the RPKM and FPKM. So RPKM is reads per kilobase of exon mapped per million fragments that's been mapped actually. And then F is actually fragments. So RPKM is used in case of a single stranded library, and FPKM is used when you have a paired sequence. So the thing is, FPKM is used so that we don't count the pairs of a particular read twice. So that's why they are distinct. And then the idea is that here you, in first step, you divide it by sequencing depth. So this sequencing depth is nothing but the total number of reads. And then generally this depth is taken in terms of per million reads. For example, you've done an Rna-Seq and you have a sample which has got around 6 million reads. So this 6 is taken as a factor. So you call it 6 as a per million factor. So you divide it with 6 and then respectively the gene length. The problem with this approach is that the total sum between of, the total sum for a given sample varies. The total sum between samples vary. So if you're taking, after you do this normalization and you take the sum across all the samples, then total amount varies. So these are not used for between sample comparisons. Whereas you do a TPM where you account for the gene length and you also account for the sequencing length, but the sequence in which it is done is actually different. First you divide it by gene length and then you divide it by sequencing length. This is like a very simple switch, but the best part is the downstream part of this is when you add all the ratio, normalized counts for the samples, they're all the same. So this allows for between sample comparison as well as it also allows for between samples and within the same sample comparison, but this does not allow for D analysis. So, but your question would be, why would you still want to normalize and why would you still want to look at this? The best, like, why would we want to look at this TPM analysis is to use these counts in case of an expression exploratory data analysis, like I said, which will be actually going through the analysis in the later part. So when you're doing an exploratory data analysis, you can't simply use the raw counts. The raw counts per se would not give, like I said, you won't be able to capture the essence of the actual differences between the groups that you're comparing when you're using a raw count. So we normalize it, and then when you do a bunch of if you want to do a PC analysis or TC or any other kind of downstream processing, so then these kinds of normalizations come into the picture. So there are, like I said, in the starting of the tutorial, I had said that we will be looking at a tool, and then that tool, I would not be taking the differential analysis in this class, but I'll be walking you through the steps of exploratory data analysis. And the tool is called as Rna-Seq, and it has been created by Michael Love and few other people. They're like the main people who have done a bunch of statistical analysis. I mean, they have given out a lot of statistical analysis for RNA-seq. So Rna-Seq is one of the most widely used a tool, and it accounts for different stuff. So the idea of Rna-Seq is to look at, for example, you have two groups. For example, there is a lot of tumors and there's a lot of control, and then the idea for it to look at, and you have several replicates within this. So the idea for it to look at is that you want to see if a particular gene between these two, groups are actually different or not. So the main thing that they do is they compare it within the same gene, so they don't have to account for what is called as the gene length. They don't have to account for the gene length, because every single gene is actually compared between these two groups, and then a null hypothesis created and everything, which I'll be taking in detail the next workshop. So so these guys have a different kind of normalization, and then which I'll be explaining in the next slide. And this normalization is called a size factor normalization. So it uses the median of all the ratios, because a median is more static, and then it is not prone to the it is not it's stable to the totally highly differentially expressed genes. So that's why this median ratios is taken. And then HR uses something called as a trimmed mean. So what they do is they take the mean value of all the genes, and then if for example, if few of the genes are very, very different and everything, they only use the particular set of genes that are they initially assume that not every gene is actually a differential expression. So they only take a bunch of genes that are that are not differentially expressed, and then they use a weighted mean for the rest of the other genes. So HR trimmed mean is actually one of those kinds of normalization. So these HR trimmed mean and the Rna-Seq medians of ratio and everything, their individual packages that is present in R does these kinds of normalization when you give it when you give the information that you give. So so these are not used in case of EDA or anything else. Yeah, so what Rna-Seq does is it does a very simple and intuitive step. So what it does is it takes the geometric mean for each gene across all the samples, and then you divide it by the geometric mean. And then, so for every gene, you take a geometric mean, and then you divide it by that, and you have a ratio. So for every single sample, across every single sample, you take the median of the ratios as your size factor. So like I said, you're taking the median of the ratios here because you don't because you actually have the median statistic itself is not very highly volatile. It's very it's like a central I mean it gives you the centrality of the data, right? So that is why this median of the ratio is taken. And size factor seems to, like, you can use size factor in EDA, but it does not have in a gene length factor. That is, gene length is not factored in it. So TPM right now seems to be the the best way to go, because FPKM is not used anymore. Not used anymore in the sense, people have stopped. Like, it's not very well used, but TPM is what generally used for exploratory data analysis and everything. So yeah, I think right now I'll actually give a bit of a tutorial, and then tell you, like, initially show, like, I'm going to assume that you have a pretty good idea about R, because entire RNA-seq analysis is mainly it's been all the codes, everything, packages are written R. So if you want to learn it, you'd have to have like very basic analysis of basic understanding of R. So R is a must if you want to do RNA-seq analysis. So in R, you would want so like I said initially, so DSeq is a package that we'll be using today to analyze the count data and the data that I have right now. So you would, beyond that, you would want several packages such as dplyr and tidyverse, because I've been using R for like some time now, and then I do most of my transformations and most of my steps using dplyr and the commands using dplyr and tidyverse. So I have to load all of them, and the command to load all of them is to call the library, and this is how you load it. Like, yeah. So there are these two other packages. It's like a package that is to pheatmap is to create heat maps, and then Arclub Brewer is a library for colors actually. So yeah, so I'm giving the directory where I have saved my files, and then yeah, so I'm reading the count data that you get after aligning to,
Hello,
aligning to a genome with any aligner. So
hello,
the
hello,
data from which I have taken this data from
hello, Akshaya.
hello.
Sorry, the interrupt. Are you sharing your screen?
Oh yeah, I am sharing my screen. I'm sorry.
But we see only
Sorry,
the screen.
sorry.
Yeah. So I
Can you see the screen right now?
yes. Now it is fine.
Studio, you're able to see, right?
Yes.
Okay, sorry. I'm really sorry. Okay, so there are a bunch of breakdowns, and this is how uh, this is how
So
the
maybe,
read count looks.
yeah, maybe you can give a quick recall for the missing part of the slides.
Oh, sorry. Yeah, sure. So the idea is that I am actually using R right now, and then the idea is that I am using a bunch of libraries that enables me to do different stuff. For example, DSeq is a library that I spoke to you earlier. It is like a library for differential expression analysis, but it also does a lot of. It's also got a lot of other basic functions to look at exploratory data analysis. So I didn't want to write a lot of functions. I just wanted to kind of introduce you people to functions that are already available. So DESeq has got few functions that we can actually use for exploratory data analysis. So I have so this is how you load the library in R. You use library and the name of the package that you want to load, and there is dplyr and tidyverse. So dplyr and tidyverse are data manipulation packages. So I have loaded both of them, and then pheatmap is a package to create heat maps, and Arclub Brewer is a package for a library for colors. So I have loaded all of these packages, and then I have set the working directory, and I have read the count file called this hammer count table, and then using the read.table command. So, and then this is how the count file looks like. So the idea is that you have different samples in the columns. So these are all samples, and these are all genes. And these are the counts, the number of, the number of reads that are present for this particular gene in this particular sample. So there are around two reads of this particular, the first 1001 in SRX102. So this is what the meaning is. So this is actually, so you might be wondering, this is actually a read count from a study that is that it used mouse, and they had two controls. One, they had a control, and then they had a bunch of samples that they have grown using some other condition, a particular condition, and they had also okay, let me, yeah, so let's look at the dimension of these counts. So it gives, there are around 29,516 genes and around 8 you have around 8 samples in this particular study. So let's also look at the metadata file. So this is actually the metadata file. So in this metadata file, you can see that there are two protocols. So first protocol is that there are there are controls. There are 4 controls, and then two, and 4 of them have been grown under different condition. I don't want to go into the details of the study because you just want to have to see that there are two groups over here. You have controls and then SNL, where let's assume that they have been grown in a particular different condition, and then you want to see if actually the cells group along because of this condition. So and also what they have done is they have taken the the RNA-seq samples at different time points as well, like like 2 months and 2 weeks. So they have taken 2 time points, and let's see how the grouping is done. Based, if the grouping is actually done, if there's a lot of for example, if the variation that is present in these samples, the variation that is captured by RNA-seq is actually mainly due to the protocol or due to time. So that is something we would want to see, like which is the main variation causing source. So that answer is given only through exploratory analysis. And if you find a lot of outlier, for example, they might all of the cells might club somewhere, and you have a very, very like totally a cell that is totally out of it's very far from rest of the points. So those kinds of cells have to be discarded from this from the analysis further down. So those kinds of stuff, everything is exploratory analysis gives you an idea about all these things. So the first thing is so what DE-seq does is it creates an object called as DDS. So here what we're doing is we are taking the data from a matrix file. So we use a function called as DE-seq dataset from matrix. So you give the count file and you give the metadata file. So the main thing about DE-seq is that you would want the column names of the count file and the row names of the metadata file to be in the same order. Okay, let's just so, and that is done using these particular commands. If you want, like I'm just printing these out. Okay, so when you look at the row names of the metadata, we can see they're in the same order as the column names of the read count. So which means that we can use the data directly. For example, if let's just assume if they have been different, for example, there's a switch, then we would actually have to reorder them by reorder them. Like I'm like you can also check if they're all same by using this command, like using the all function, or you can also use this something called as all dot equal as well. So you can check if all of them are in the same direction, and if they're true, you can proceed with calling the DDS. So this creates a data object. For example, it's got several. So the main thing is that here we have something called as design. So in design, the thing is that, so for example, if you're doing a exploratory data analysis, you would not want to give any information. So the information per se over here is to, you're taking the metadata file, but this particular information is only used for annotation. But in case you want to do a differential expressions and this differential expression analysis, and you want to give, you want to read the difference across the protocols. So you want to see if there are genes that are upregulated or downregulated with respect to the control samples and the treated. Let's call it as treated. So if you want to see the difference between them, and that is something that has to be captured in the design. So you would, so if you want to look at that, you would give the design matrix as protocol. So you would, instead of design being one here, you would call it as, like you would say, protocol over here. But for exploratory data analysis, you would not want to do any assumption. So without any assumption, you are going to look at the data and see which, like you want to see here, there are two varying sources, one being the protocol and the other being time week at which this is the samples were sequenced. So we would want to see which is the main variation causing source. So that's what we are going to do right now. So to not give any presumption, you just put them design matrix as one. So like initially we had looked at the size factor normalization, right? So this is what size factor normalization. You can get the size factor normalized data using this particular function. So okay, let me show you how well it. Sorry. Yeah. These are the size factors that has been calculated for each of the samples. And when you look at the plots, let me show the plots. Like we said before, the size factor does not account the gene length, but it accounts for the total number of counts that are actually present. So this is actually kind of, size factors are actually proportional to the total number of counts that are present. So they kind of normalize for the RNA composition, that is nothing but the differential expression, and they also account for the total number of reads that are present. So it's just a plot to showcase that. So we take the normalized counts. So these counts are size factor normalized. We can also use size factor normalization counts. So, but then we would also want to, using the size factor normalization counts for the exploratory data analysis, we can simply do a hierarchical clustering based on the distance that is present in each of these samples. So a very simple distance matrix in the dist. Here over here we are using a function called as dist, and this particular dist function uses the Euclidean distance actually over here. And then we create a hierarchical clustering. So first we'll see what kind of clustering is done over here. So when you look at the cluster dendrogram, you can see that the controls seem to be, under a totally different branch, like main two branches, and then controls seem to be in a single branch, and the rest of the SNL, treated samples are like the treated samples are under a different branch. So, so this is with respect to the protocol. And if you also want to look at the variation, like the time bearing the variation, so you can kind of see there's an inner cluster, and the inner cluster is like within the control and the the treated samples. So within this particular treated sample, we can see that 2-month samples are more closely together than the 2-week samples. So the idea is that this ETA is very intuitive. So this is something you would normally expect as well. Like when you're doing an RNA-seq, when you're differing with different conditions, so these cells will capture the different conditions, as well as they will also capture for how long this particular treatment has been done to them. So these are like, it gives us an it gives us an added validation that what we have done, like the samples are actually the correct samples as well. They're not like there's no intermixing or there's cross contamination or anything like that. These are like, so EDA is like the very basic step that you want to be really sure about before going through any other kind of analysis. So, and then
Hello, Akshay. Sorry for the
yeah,
interruption. So would you like to take some questions from the comment box? Like,
yeah, sure. Yeah.
so there
Yeah, let me go through the questions. Should I go through them one by one, or
maybe you can go with Sriram. He asked like this table has a genes or transcripts as a rows. So maybe you can go with this one. And
okay, the thing is that this particular table has got the genes. These are actually the genes with respect to rats. So these are not transcripts. These are rats. But if you're in case using transcripts, you would treat them in the same way. So these are genes. But if, for example, if there are transcripts, then you would have ENS. So instead of S, you'll have ENT, and some number. So that would be in the rows, and then the samples in the columns. So here the answer to your question is these are genes right here. So depending upon your alignment, like if you're using a particular transcriptome specific alignment, then you would have reads pertaining to those transcripts. But then the thing is that you treat them the same way. Yeah. Any other question? Let me.
yeah, another one is like from Jessara, like so we can compare the control and test groups via R. Also, will R Studio also normalize it for us? I don't understand the question, but hope you can
So
understand.
will R Studio or?
So the question from Jessara, so we can compare the control and test groups via R.
Yeah.
Also, will R Studio normalize it for us? The question was
No, not like that. So you have several packages to do that for you. For example, or you can write your own function to do that if you know the steps to normalize it. For example, in DSeq, like I said, you have this particular function called as estimate size factors to do this, the specific steps to normalize your reads. Or for example, if you're doing a RPKM or for a TPM, you would know you would know your library size. You would know your the total number of reads from for example, if you're using SAMtools FlagsStat, you would know the total number of reads, and then you could use that particular number to divided by the total number of per million factor that I spoke about. And then, for example, the gene length, you would have to take the information from Ensemble or wherever you're taking your data, I mean, reference data or your annotation data from. So you would have to use information from those files to do these kinds of normalization. For example, if it's a simple logarithmic transformation, you simply add a pseudo count and then do a log transformation. So that also works, but you have specific packages for in case of DSeq, and then there's R as well. So those specific packages can be used, or you can write your own code. I mean, you can write your own simple function for it. Yeah.
Yes, another one from Sriram itself. It's will having transcript or genes change downstream analysis?
See, it will, because to be honest, for example, what you do in downstream analysis is to look at pathways that are actually being changed, right? So if you want to look at if your end analysis is to look at pathways that are changed, or pathways that are, for example, upregulated in a tumor cell or downregulated in a tumor cell, you would definitely want to stick with genes, because you already have several gene sets that pertain to a particular pathway. But if you're looking at differential expression of transcripts, where you say that, okay, until, like, generally these transcripts are present everywhere, and you want to look at transcripts that are only expressed in a specific condition, like, then you would want to go with transcript analysis. So yeah, then you would want to, like, for example, okay, in a normal state, for example, for this particular gene this transcript is expressed, but in tumor cell this particular transcript is expressed. But to do further downstream, you can probably look at few of the networks, and you want to look at how these transcripts are behaving like a group. You could do that, but you cannot per se find out the pathways in which it are. Like, what you can, another way what you can do is, what you can do is take the transcripts, and then so all these transcripts are for a particular gene, right? So you can convert these IDs into gene IDs, and then probably look it up like that. So, but the thing is that for a particular gene, there might be several transcripts that have been expressed. So again, you would have to collapse those reads and then go down with the analysis. So you can actually look at it.
Okay. So there is another question from Bhavani. So how effective can RNA sequence be used for fine needle aspirant sample?
Yes. So yeah. So fine needle aspirant samples, you mean like fine needle biopsy, right? So if they're like fine needle biopsy as well, you have enough sample, you can definitely. That all depends upon, like, for example, if it is a fine needle aspirant, from my experience, you're actually taking a biopsy of a sample, which means there is some diseased condition, right? So then you would have to talk with the pathologist. He would require amount of sample, and then he would give you an amount of sample. So if the needle aspiration itself has got very little amount of sample, then you would not be able to do that. So it depends on how much sample that they have got, and if you're very lucky, you're a researcher, and then the luck is in your side. You might have a lot of sample, and then they might give you enough for your RNA-seq. So yeah, so all of that depends upon the amount of sample that you get, and also depends upon the pathologist. If they I've heard that very old pathologists, I mean, like, people who have a lot of you know um, a lot of experience are like, they'll actually give you samples, but the newbies are kind of reluctant to give you anything. So I've heard it from other people. Yeah, so that's my take on it.
Okay, hope that convince Ms. Bhavani. Yeah.
So that's right. Do we should I proceed with the? There's only two more.
So Akshaya, it's
Yeah.
almost 1 hour. So how much more time you need to for today's session?
So there's only one thing I want to discuss. Like, I want to discuss something called this transformation. How these just give me 5 minutes. I'll be done with it.
Okay.
So
Go
yeah.
ahead.
So the thing is, for example, you're doing some normalization so that your normalized reads are actually, you can, for example, if you want to look at in a PCA plot. So so the main problem with respect to RNA-seq is that okay, no, it's taking a lot of time to plot. Okay, must have the. I'm sorry, my computer is terribly slow. So the
Not an issue.
so
Take your time.
Not loading open. Okay, yeah, so I've got the plot here. So the idea is that these so when you look at RNA-seq counts, so what I've done is I have log normalized the counts of one sample. This is the first sample and the second sample, and I've plotted it in a logarithmic scale. So what you can see is that when when the counts are very high, for example, you have very high number of counts, the variation of these counts in different samples is very less. Whereas when you look at very low counts, there's a huge variation. So the problem with this is the problem is that you might falsely, by mistake, you might consider these as like these genes over here as a differently expressed gene. So what we have is there are several transformations that you could do so that these are not considered as actual differently expressed gene. So one of the main transformation for that is called as a variant stabilizing transformation. So probably I can take the like the steps with which how it's done actually, but I'm just showing a plot how different this makes like okay see so this is a log transformed count, but where you can see there's a very huge variation when the counts are very less. But when you do a VST, there is nothing but a variance stabilizing transformation. You can see these variants being accounted for, and then they're actually rectified to reduce the they kind of pull down next to the mean of the line. So these kinds of transformations are very important when you're actually wanting to do any exploratory data analysis, because if you simply use normalized counts, actually this data is very good. You can actually see only with normalized counts, you're just normalizing the counts and you're trying to do a bunch of hierarchical clustering, and you're able to see the groups that you want to see. But in most cases, this does not happen. So what you would want to do is apply the transformations. For example, you have a variance stabilizing transformation, or you have a regularized logarithmic transformation, or a simple log as well. But the problem with simple logarithm transformation is that it does not rectify the variance that is present at low counts. But regularized logarithmic transformation and these transformations are helpful in that sense. So when you look at heat maps using these kind of transformation, you can see very clearly you can, for example, when you look at PCA, so there's a function for PCA and DE-seq itself. It's called product per CA, and then we can simply give the variable that you want to color it. For example, here we're looking at protocol. So, and we can very well see that it's got like, so like I said initially, we want to see which kind of variable is giving the biggest there are, so in the metadata we looked at two different variables, right? One being the protocol and the other being the treatment time. So here we can see that in case of PC1, that is able to capture around 93% of variance, there are two different groups, and these groups pertain to the control and the treated group. So it's very clear that the biggest variation that is captured over here is actually, it's captured by the protocol difference itself, that is in which the cells are actually grown. So PCA is very intuitive in that sense, PCA, and we can also use PSNE, and then there are several other actually functions to use to do exploratory data analysis. So this is what I had. Yeah, this is the end of my workshop. Yeah. So probably next week we can look at differential analysis and a little bit of pathway analysis. Any questions you could ask me right now?
Yeah, so guys, we'll take 5 more minutes for questions. So if you have any question, you can ask.
Yes, sir. How effective can Rna-Seq be used for?
It's already a trust, I guess.
How much sample is required, minimum required to do Rna-Seq analysis? I am not sure, Sumitra. I have no clue because I am not involved in that processing and everything. I have no clue. It's probably a biotechnology. I mean, like where I get from my samples from, it's given by postdoc, and then he does a lot of I mean, he knows how much is required. I'm sorry, I don't know about that. So yeah, so end of my
So are there any questions, guys? If any, please unmute and you can ask it.
okay.
Okay, it seems like there are no questions. So
Yeah.
what we do, like maybe we can give a small introduction, like what you are going to present for the next session. Then
So
it will.
I will be talking about differential analysis per se, like how D-Seq handles it. Like, so there might be a lot of false positives and everything, right? What statistically, what D-Seq does. Like, if people are interested in the statistical viewpoint, I could take that. Like, if you guys give me an idea of in which viewpoint you want, like, you just want me to do a bunch of analysis and show you like a tutorial, that would be fine. Or you would want me to go into the statistics and tell you how different what it actually does. But that's a lot of theory. If you want that, I could do that. So that's a feedback that I want from you people. Yeah, so my plan was to go through